The protein. It is considered as the building block of life, and that is, it is found in every cell of the body. By far, it is the most important nutritional molecule or a biomolecule of a biological compound. In fact, it needs no further forward, as it is derived from the Greek word proteios, meaning it is of primary importance. Protein is a linear polymer that is built on a monomer called the amino acids. As below the word I mentioned is an illustration of a monomer called amino acid, or what we call the single unit of a protein. Now, at both sides are multiple product of peptide bonds, that is, bonded, complex, and dedicated structure of polypeptide chain. And as for the sources of protein, protein emanates from an animal sources, plant sources, and even soy products. If it is from an animal sources, it is said to be a complete protein source. And this may be from chicken, beef, fish, and dairy products. If it is from a plant source, it can be described as an incomplete protein source. This can be from beans, lentils, nuts, and whole grains. However, soy products may be derived from quinoa, that is similar to the rice, and seed of leafy green plants. The protein functions. Protein has a multiple cardinal functions of prime importance as a structural function. Literally, when you look at a person at the right clip, you are looking at her proteins, because her body muscle, her skin, hair, and even the nails are made up of protein. In signaling, from the pituitary master gland to endocrine and exocrine glands, hormone keeps metabolism running. Protein as a catalyst. Enzyme hastens and speed up the human biochemistry to a millionfold speed, and that's made up of proteins. In immunity, from the foreign antigen etiologic invaders to human defenses, that's the color blue. They are the antibodies, and they are of protein composition. In regulation, from the DNA expression of genes, the perpetuation of species are being made possible, and that is with the magnificent role of protein. And even the most toxic venom of a poisonous snake is being synthesized from a derivative of protein. And in transport, the hemoglobin from the red blood cell is what you're seeing at the right. And it kicks off as a carrier of oxygen from the intravascular to intracellular, out to alveolar, respiratory physiology. And finally, protein as a storage. Ferritin from the liver stores iron, and casein are being stored in milk. and even the oval albumin that is stored in eggs. It is being preserved. Indeed, protein is a building block of life more than anything. In this era of today's generation, the world is in dire need of a medical expert and a scientist that will safeguard the humanity's health and well-being. The world is being threatened either by a genetic mutation that leads to either oncological nature of diseases, and notwithstanding the existence of the pandemic that causes havoc to all nations. And if you want to become a protein biochemist, and not only for the purpose of high grade, then with all your might, you must know everything about the most important biomolecule, the most striking and the most spectacular amino acid. You must be able to know their names, structure, the letter code, whether it is a 3 or a 1, the ionization process, the hydrophobicity, the hydrogen bonding properties, and other chemical properties of amino acid. We can classify amino acid as to, one, the charges, whether they're electrically charges, positive or negative. Another classification is based on their polarity. And another classification is based on whether they are hydrophobic or hydrophilic. And there are some special cases. Now, hydrophobic amino acids are composed of 8, and they are the A, B, I, L, M, F, Y, and W. That's to name it on a one-letter code. Now, hydrophobic amino acids has a carbon-rich chain, and it does not interact with water, as you may see in the clip above. The waters are being repelled from the outside. They are water-hating. They are like tiny drops of oil. They want to get out of water, and they try to condense in the middle. Yes, you may notice the hydrogen bonding and the polar grouping. They interact to stabilize the in and out structure of the protein. Now, the hydrophilic amino acids. The hydrophilic or the polar amino acids are composed of 4 amino acids. They are the S, T, N, and Q, and G, rather. The hydrophilic amino acids are said to be water-loving, and they interact with the water. As you may look at the red molecules, they are being attached to the central carbon atoms. And these are the partially charged, which seems to be protonated form. And when they are fully charged, they are in a deprotonated form. And mostly, they thrive in an acidic environment. They are charged, they interact with the opposite. And amino acids can be classified as according to their charge. And the charges are separated into positive and negative. In fact, there are 5 of the charged amino acid. The 3 of them are said to be positive, the arginine, histidine, and the lysine, and they are described as basic in nature. And the 2 are negative, which is the aspartic and the glutamic, and they are said to be acidic in nature. As you may notice in the clip, there is a positive side of the hydrogen and negative side of the water, and that the charge will interact with the opposite charge of other amino acid or other molecules. Finally, we can classify amino acid as aliphatic or aromatic compounds. If it is aliphatic, there are 5 of them. And when we define aliphatic, they are composed of a straight chain. If you may look at the picture, the carbon atoms are appearing in a straight chain branch or cyclic compound, and this can be saturated or unsaturated. And looking at the right side, if the amino acid is aromatic in nature, then they can be described as a compound with one or more conjugated, and that the benzene ring or heterocyclic ring can be seen as it is circular composition of carbon atoms as having a ring structure. The amino acids as the building blocks of protein. There are more than 700 amino acids, and they occur naturally. However, in the human body, there are only 20 that are identified. And at the left side, there is a list of amino acids based on their chemical composition. This amino acid can best be represented by using a 3-letter code, which will play an important role in our discussion of synthesis of amino acids. Together, much more, it will be easier if the amino acids can be identified by a 1-letter code. Now, out of the 20 amino acids, 9 are identified as essential. Essential in a way that they must be consumed and must become a part of a human diet. 5 are identified as non-essential in the way that the body can synthesize it or the body can produce it. Now, the remaining 6 are said to be conditional, and they may become essential only at a certain life stages, or they may not be there in a certain disease state or condition. In our discussion of the building blocks of protein, the amino acid, the need to rigorously and rigidly recall the basic chemistry is paramount. And in order to ease up or simplify our succeeding discussion, there are important concepts that we have to revisit and look back from our organic chemistry. As from your old notes and from your basic chemistry discussions from K-12, you might have learned that there is a special nature of carbon and hydrocarbons in the formation of biomolecular skeleton. And during your early encounter in chemistry, you might have taken emphasis on what we call the functional group, its properties and reactivities. Now, it is at this note that we will connect the importance of carbons and the functional group in our later discussion. The carbon. Organic chemistry has been described by chemists as the chemistry of carbon. I mean, the whole branch of chemistry is set aside just to study the carbon compounds. That is organic chemistry, as differentiated from inorganic chemistry. Now, carbon is the main element in organic compounds, and so carbon is essential to life on Earth. For without a carbon, life as we know could not exist. In our succeeding discussion, you may take into account that the appearance of every single monomer of amino acid is composed of a carbon or a multiple of hydrocarbons. Let's take a look and a listen at some selected samples of amino acid. At the lower left is a simple monomer of amino acid, the glycine. You may notice that in the middle, there is a backbone two chain of a black balls of carbon. Let's take a look at the middle. In the middle, the monomer tyrosine of amino acid has a middle ring chain, circular ring chain, and above are all composed of a black carbon balls. And at the right side, you may notice that there is a long vertical chain of a carbon in the middle, and that's called the hydrocarbons, that serves as a backbone of the amino acid. Much more, if you will look at the clip up left, it displays that a protein, whether a polymers or a polypeptide complex protein, is being made up of a carbon skeleton or a carbon base backbone. The functional group. It is an important concept of organic chemistry, as mentioned in the previous slide. And so as in our amino acid or protein building blocks discussion right now. It is the characteristics group of any organic molecule. As you may notice, there are multiple formed chemical arrangement or a compound arrangement. They're like prototype, a combined group, and they are separated as being encircled. Literally, they are like an ingredient in a cooking menu that when you mix and match, voila, you can form a substance. Now, shown here are 40 functional groups. In as far as amino acid composition is involved, we will only highlight two of them. They are the amine group and the carboxylic group. Now, when you combine them together, dear students, this is how an amino acid chemical structure will look like. A combination of an amine group at the left, represented by H3N positive, and a carboxylic group at the right, represented by a carbon and a double oxygen negative. And this is the ball and stick appearance of amino acid. The amino acid chemical structure. As previously illustrated, the amino acid chemical structure appears to be composed of carbon or the alpha carbon as its center, playing as a backbone or skeleton of the amino acid molecule. It is so called as the alpha carbon because of its proximity to the carboxylic group. Added to that, the carboxylic group is at the right side having a negative charge, and at the left side is the amine group. On the upper side chain is the hydrogen, and the R group as the lower side chain, which later on will play an important role in the identification of amino acid. Now, amino acid is basically known as sweeter ions. Sweeter ions from the German word sweeter, meaning it is a hybrid. It is a hybrid because this is a molecule or an ion having a separate positive on the left side and negative charge group on the right side. That means it translates to the word dipolar. Added to that, amino acid is an ampholytes. and ampolites because it can act as either an acid or a base. And finally, amino acid is in a solid state when in an outside external environment. Like when you put the amino acid into water, it will be dissolved. And much more inside the human body. It will be subjected to multiple physiological pH range. And both the carboxylic and the amino group will completely ionize and react. And this is to be discussed in the next slide. The structural, the chemical, and the identity changes of the amino acids. As from our previous discussion, we have learned that the amino acid chemical composition is made from the carbon as the central backbone of a molecule. And at the right side, we have the carboxylic group. And at the left side, we have the amine group. And the upper side chain is represented by hydrogen. And the most important is the R group, from which the R group represents the R or the rest of the chemical that will be added to the amino acid basic composition. Now, varying the R group would mean changing the structure, changing the chemical, and changing the identity of the amino acid, from which the changes in the R group will be derived and taken from the functional group. The functional group that we have mentioned in our earlier discussion. Now, changing the R group with any member of the functional group may be likened to the analogy that we will present in the next slide. In this slide, as we change the R group from the amino acid basic chemical composition, it is as if we are changing the character of a person, and that is synonymous with the changes in chemical reaction. As we keep on changing the hat of person, the person's identity is being subject to change, and that a new character will always emerge as we replace the R group in our amino acid. Now, from these changes, we can identify several amino acids, and we have named the amino acids in the body as 20. Now, here are some of the representation of the different amino acid and their structural and chemical changes. Let us try one. Changing and varying the R. First, we have replaced the R with a chemical structure that will lead us to asparagine. Another chemical structure that replaces the R represents the threonine. Another will lead to proline, and another will lead to leucine, and another will lead to alanine, and another will be identified as valine, and another will be identified as methionine. And the simplest of them all will be the glycine, which represent an additional replacement of R group as hydrogen.